Okay, let's take a quick look at eukaryotic gene regulation. So how eukaryotes can regulate and control gene expression. When we're looking at regulating gene expression, we're talking about turning genes on and turning genes off. So again, we don't want to be making proteins all of the time. We just want to be making them as needed, and different cells are going to require different proteins to be produced. And so we want to turn genes on and off in the different cells. We're going to be using those proteins that are necessary for that particular cell's role and only that particular cell's role. So you're turning genes on and off. And so that's differential gene expression when you're turning certain genes on in some cells and genes off in other cells. So not all cells are expressing all of the proteins all of the time. Okay. So really what you're doing is you've got a whole bunch of genes and they're going to be producing proteins, but they're not going to be producing the proteins all the time in all of the cells. And so we're turning genes on and off as needed in those particular cells. So transcription factors are going to be signals that are going to be responding to hormone signals that will turn the genes on and off. So there's cell communication that helps to turn on and off the genes in those cells. When we look at the stages of gene expression and protein synthesis, each of those steps along the way, that whole process, are areas where gene regulation can be controlled. So we're kind of going to look from the inside of the nucleus out, from the DNA to the protein, and how it can be regulated at each of those different steps. So again, starting in the nucleus, that's where you've got your DNA, and that's where transcription will take place, and then translation takes place in the cytoplasm. But every single step along the way is where gene regulation can take place. So if we look at the DNA inside of the nucleus, we can look at how the chromatin structure can be regulated. So we know that DNA is coiled around proteins called histones. When those proteins, when the DNA is wrapped tightly around those proteins, then it's very hard for gene expression to take place. So it needs to unwind in order for the genes to be expressed. So when they're highly packed, it's called heterochromatin. And in those areas, then the DNA is not going to be read. So there's not going to be any gene expression. If we modify those histones, the proteins that the DNA is wrapped around, that can alter the gene expression as well. So histone modification is adding molecules to that histone that will cause it to either wind tightly or wind loosely around the histones. So adding a methyl group, and that process is called methylation, that's going to condense the chromatin, and it's going to make it tighter and harder for transcription to take place. And if we add a phosphate group to the histone, then that is going to cause it to loosen, and then it's going to be easier to to do transcription. So methylation causes it to wind tightly, and phosphorylation causes it to be loosely bound. So you can see in the picture, you've got the histones in the purple, and then the DNA is here in blue, and they're wrapping around there. So adding the methyl group to the histones is going to wind it tightly, and it's not going to be able to do transcription. And then the acetylation is going to wind it loosely. So two processes that can cause it to either be wound tightly or wound loosely. Okay. So that's the histones. We can also add molecules to the DNA that's going to cause it to be able to do transcription or not do transcription. DNA methylation is the addition of those methyl groups, and that's going to cause the DNA to become inactivated, and so it's not going to be able to do transcription. And that leads to genomic imprinting, and in some cases that methylation can be passed from one generation to the next, basically turning off genes. Even though you have the DNA coding for certain proteins, in genomic imprinting, you're basically shutting that process down, that you're shutting down that gene, so that's not being able to be passed down to the next generation. And that's what genomic imprinting is. So DNA methylation is turning off genes. And epigenetic inheritance is that inheritance of that genomic imprinting and that DNA methylation. Chromatin modifying enzymes can also affect transcription. And so those chromatin modifying enzymes are going to provide initial control of gene expression by making certain regions of the DNA either more or less able to get the transcription factors and the transcription machinery to bind onto it. And so that could either enhance or it could slow down transcription. So in eukaryotic cells, there are control elements that are segments of the non-coding DNA that are going to serve as binding sites for transcription factors that help to regulate transcription. And the control elements and the transcription factors are going to bind, and they're critical to the precise regulation of gene expression in different cell types. So the control elements really help the transcription factors bind on and make the whole process more efficient. So if we look at this picture here, you've got a sequence of DNA, and in that DNA, you've got your promoter region, you've got your exons, your introns. So this is the portion of the cell that's going to produce the protein, the messenger RNA, which would then produce the protein. Upstream of it, so in another location on the same chromosome, is what's called an enhancer region. That enhancer region is going to help the transcription factors bind on to the transcription site so that the RNA polymerase can bind and then transcription can take place. So the primary transcript is going to be made from this portion of the DNA with the enhancer region upstream from that. Okay, so the transcription factors, again, are going to be proteins that help that RNA polymerase bind onto the promoter region so that transcription can take place. And the control elements are going to help those transcription factors bind on to that initiation site. So the enhancers are going to be molecules or portions of the gene over here that will then help bind those transcription factors. So they're going to basically activate or turn on those genes. So they're going to make that process of transcription more efficient, a much faster process. So if we look at this picture here, our enhancer region is over here, and then that DNA molecule kind of folds over, and that enhancer region is going to help those transcription factors bind on. So we've got these mediator proteins that help the transcription factors to connect, which then makes the RNA polymerase able to bind on and more efficiently do transcription, making the messenger RNA much faster. So the control elements are going to activate transcription, but only when the appropriate activator proteins are present in the cell. So not all activators are present at all times. And so it's not going to fold over and do transcription all of the time, only when those activator proteins are present in the cytoplasm. So different cells are going to have different activators present in the cell. And certain cells are going to have certain activators present at certain times. So, for example, the liver cell is going to be making enzymes like insulin and things that are going to be secreted from the cell. They're going to be producing things that the liver, sorry, not insulin, but liver is going to be producing a whole bunch of secretory enzymes that need to be produced. And one example is the crystalline gene is producing molecules that the liver needs. And so you're going to have these activators that are present that are going to cause that particular gene to be expressed in the liver. But in the lens cell, the lens of your eye needs to be clear. You need to be able to see out of it. You don't want albumin being produced. You want other proteins to be produced in the lens. We want it to be nice and clear so that we can see out of our lens. And so there are going to be different activators available and present in that cell that are producing different proteins, but not the ones that are going to be produced in the liver. So you're going to have different proteins being expressed because you've got different activators present in the cell that are enhancing that transcription process. So here it's showing you, you've got all of these genes in your cell, but in some cells, you've got the enhancers and the control elements, but you don't have the activators in all of the cells. So the liver cell is going to have the activator available for the albumin gene, but the lens cell is going to have the activators present for the crystalline gene so that it can make the crystalline protein necessary for a clear lens in your eyeball. So different activators are going to be present in different cells. So different switches are going to be turned on in those different cells. Okay. So that's kind of looking at the switches. So gene switches turn the switches on and turn the switches off. Then when we look at how chromatin is folded inside, when the chromosomes are in the nucleus and they're unfolded in the form of chromatin, that DNA is kind of folded up in a unique pattern, and how the DNA kind of loops in there is also going to be a mechanism for controlling the production of messenger RNA. So RNA is going to be produced in certain areas, and so you're going to see loops of chromatin in certain sites within the nucleus, and those are going to have specialized functions. So if you look at this picture, you kind of have these different color-coded regions, and they're all kind of functioning together. So that's another step of how we can control and regulate gene expression. Okay, moving further, now we're looking at post-transcriptional regulation. So after the messenger RNA has been made, we can regulate that step as well. So we're looking at, again, every single step in the transcription translation process is a way that we can turn genes on and turn genes off. So looking at that RNA, and we talked about how RNA is processed and how we can splice out the introns and connect together the exons. Alternative RNA splicing is another way of producing different proteins and therefore regulating gene expression. So alternative RNA splicing is cutting and pasting the messenger RNA molecule in different ways to produce different proteins. And so you can get a whole bunch of different proteins from one section of DNA. So one gene could produce different proteins depending on how you cut it and paste it together. So DNA here, if we look at our DNA, we've got exons 1, 2, 3, 4, and 5. If we make that primary transcript in that entire thing, and we're transcribing it all, we can cut and paste and put together 1, 2, 3, 4, and 5 in different ways. You can take all of those 1, 2, 3, 4, and 5 exons and put them together and make one protein. But if you take 1, 2, 3, and 5, you're going to get a different protein. Or if you take sections 1, 2, 4, and 5, you're going to get another different protein. So how we cut and paste and put together those exons, we can actually get different proteins being produced. And that process is called alternative RNA splicing. Also looking at the messenger RNA, how long the messenger RNA sticks around is another step in how we can control gene expression. So the messenger RNA is going to have a certain lifespan. It's going to stick around for a certain amount of time. And so the longer it sticks around, the more proteins that you're going to have being made. So if we control how long it sticks around, that's another way of controlling gene expression. So there are untranslated regions of that messenger RNA, sections that don't actually code for proteins. How long that section is going to have an influence on how long that messenger RNA survives in the nucleus. And so we can regulate gene expression by regulating that length of that untranslated region in the messenger RNA. So controlling the lifespan of the messenger RNA is another way we can regulate gene expression. Then initiation of translation is, so now we're moving into translation. So from transcription to translation, we can also control translation. So we can control the initiation or the start of translation. And so if it starts right away, then you're going to be producing a whole bunch of proteins. But if you block that translation, then you're not going to be making the protein. So blocking regulatory proteins that bind onto the messenger RNA is going to stop translation and therefore stop protein production. So we can regulate and turn on and off translation as well with various enzymes and regulatory proteins in the cytoplasm. There are also proteasomes, which are protein complexes that are going to, after translation has taken place, they are going to be cutting the proteins. And so how you cut them can also regulate gene expression, and we can then control the protein production. And so the proteasomes are going to be those protein complexes that bind to the protein molecules and break them down. So how long the proteins stick around in the cytoplasm is also another way of controlling gene expression. So here it's showing you a protein that's going to be degraded. Ubiquitin is a protein that would then bind to it. So basically, when we want to get rid of a certain protein, we can add that ubiquitin, and that's going to tag it so that it knows that it needs to be destroyed. It goes into the proteasome, and it basically chops it up and recycles all of those pieces. So how long the proteins stick around in the cytoplasm is another way of controlling gene expression. So really, you are looking at all of the stages of protein synthesis and each area in which we are regulating and controlling the proteins that are being produced and sticking around in the cytoplasm. So we can modify the chromatin, the DNA in the nucleus. We can modify transcription. We can then also modify the RNA that's being produced, so RNA processing. When the RNA leaves the nucleus, we can then regulate translation and how long the messenger RNA sticks around. And we can also, after translation has taken place, we can modify the proteins and we can regulate the proteins, how long they stick around. So basically, every single step of protein synthesis is a way that eukaryotic cells can regulate gene expression. Let me know if you have any questions.